Our project explores using Hdc Gal4 hybrid genes to identify cells expressing Hdc and containing histamine in Drosophila melanogaster. The authors of this project are Annabelle Mag, Catherine Westphal, and Judith Ismaya. Our mentor is Martin Berg, PhD, and this research was done through the Department of Biomedical Sciences at Grand Valley State University. One central question in regulation of gene expression concerns how tissue and cell specificity of expression is regulated. Gene expression is essential to understanding and analyzing the results of our research. Gene expression describes how genes encode for proteins, and these proteins control cell function. The cell's double-stranded DNA is used to produce single-stranded RNA through transcription. Next, the RNA is spliced to produce multiple mRNA products, and this mRNA is exported from the nucleus of the cell. Each of the mRNA molecules would then produce a protein through translation.
So what characteristics of a gene should we look to study? We can look to see, is the gene expressed in multiple types of tissue? Is the gene expressed during various stages of development? Are there multiple forms of a gene's mRNA? These differences could include multiple 5' promoter regions, multiple 3' end regions, or multiple exons. It is easier to study a gene when it affects a small number of cells. We will be studying the HDC gene, which encodes the enzyme histidine decarboxylase. We have a few different potential approaches to study gene expression. One option is tissue in situ hybridization to localize specific mRNA. In our case, there is not sufficient differences between mRNAs to utilize this method. We could also use promoter or gene fusions with GFP, and this has been done before. However, the fusions do not discriminate between the 3' ends and the 5' ends, so it will not be useful for our purposes. Another method is enhancer trap labeling using Gal4 UAS-GFP, but this approach has not worked thus far for HDC. Finally, we can use mRNA 3' labeling using the Gal4 gene, and this approach is what we took to study HDC gene expression. An important question to ask is, what is Gal4? Gal4 is a transcriptional activator from yeast that activates the UAS DNA sequence. The Gal4 protein binds to the UAS DNA sequence and acts as a switch to turn genes on and off. In our study, we used a UAS-GFP reporter gene to identify cells expressing Gal4, which will label specific mRNAs being expressed. The HDC gene has been predicted to generate 4 distinct mRNA molecules. The blue bar at the top of the figure represents the HDC gene region. Beneath it are the 4 mRNA molecules generated, each with different transcripts. We can label these mRNAs as HDCRA, HDCRB, HDCRC, and HDCRD. These transcripts are regulated in the nucleus where transcription occurs. So now we ask, why are these mRNA transcripts produced, and is there a difference in where in the fly they are located? Recent molecular genetic tools have been developed that allow us to visualize mRNA expression. Here we can see the 4 different mRNA patterns and where the Gal4 gene is inserted. There are 2 different types of marked transcripts, one with Gal4 at the end of the RA, RB, and RD transcripts, and another with Gal4 inserted at the end of the RC transcript. As there are 2 separately labeled HDC Gal4 genes, it may be possible to identify histamine in the cells using any one of the 2 major types of mRNA molecules. So our hypothesis is, can specific HDC mRNA transcripts be expressed in neurons that are non-overlapping? To answer this, we need to identify cells in flies that express Gal4 under the control of the HDC gene. We can do this by utilizing 2 different HDC Gal4 hybrid genes and use Green Fluorescent Protein, or GFP, to visualize expression. We also must ask, where is HDC expressed in the central nervous system? And we can detect HDC expression using histamine immunolocalization. This image provides reference as to where histamine should be located in the larval central nervous system. This image was taken using confocal microscopy after the larval central nervous system was incubated with histamine antibody that detects HDC-dependent immunoreactivity. Histamine is detected with red fluorescence. There are 2 brain hemispheres, and we can identify 4 cells in the brain with histamine, pointed out with the arrows. Here we can see the ventral ganglion, in which there are 10 cells with histamine presence. For the purposes of our research, before we can analyze the presence of histamine, we need to understand whether or not GFP is being expressed in these cells. In our study, we used a cross to produce flies using Hdc Gal4 hybrid genes to express GFP. On the left is the male fly and on the right is the female fly, and these flies produced our cross. On the left again, we have the male fly, and we see that Hdc Gal4 interacts with the UAS-GFP by binding, represented by the arrow. This induces green fluorescent protein, also known as GFP, which we can detect with microscopy. We also see Gal4 binding to the UAS, with the female fly shown here on the right, exhibiting expression of GFP.
To get our results, we dissected and examined brains from the flies in the larval stage and did live imaging using epifluorescent microscopy. After this initial screening using the epifluorescent microscopy to determine if GFP was expressed, live imaging using laser scanning confocal microscopy was done on the larval brains. We then did colocalization of histamine and GFP expression in the larval brain, where we imaged the tissue using confocal microscopy. These first 2 images are bright field and GFP fluorescent combined images of the larval CNS, each fly representing one of the 2 different Hdc Gal4 genes. The bottom 2 images are just the GFP fluorescent image. These are the Hdc RA-RB-RD images, and these are the Hdc Gal4-RC images. The living tissue between these 2 larval brains appear to be similar. The closeness of these images may be due to it being difficult to detect all cells in the live tissues. We really can only see a few brain neurons in the hemispheres, and 10 cells in the ventral ganglion for both specimens. Based on our live imaging analysis, we found that Hdc Gal4 hybrid genes do induce expression in cells, which are similar in our 2 different labeled genes. However, the resolution for our entire CNS is poor in the live images. Therefore, we may not be seeing expression in all possible cells. A question that still needs to be addressed is, do GFP expressing cells also contain histamine? We further examined larval brains for GFP expression indicated by green fluorescence and histamine by using histamine antibody to image histamine-like immunoreactivity indicated by the red fluorescence. These next images are the larval brain images we took of each of the 2 different Hdc Gal4 hybrid genes after they have been put in fixative, which allows for better resolution and shows individual cells when we use confocal microscopy to scan the images. These first set of images are the bright field images. These are the GFP images, where we can now see a difference in the GFP expression in the Hdc Gal4 RA-RB-RD fly and the Hdc Gal4-RC fly. In both larval brains, we can see GFP expression within the hemispheres, as well as 10 cells within the ventral ganglion. The GFP expression here is very similar to GFP expression in the live images. Next are the images showing histamine-like immunoreactivity. An important thing to notice is there does appear to be histamine in cells for both the brains. However, histamine appears to be located in different cells between the 2 brains. For example, histamine appears to be in the cells for the 2 hemispheres and the ventral ganglion in this fly, But we don't see histamine in the ventral ganglion for this fly, and we do see it only in the hemispheres. And here is the combined image of the GFP and histamine-like immunoreactivity. What we can take away from this is that there is a difference between the expression of GFP and histamine-like immunoreactivity between the two different Hdc Gal4 hybrid genes. These are zoomed-in images of the ones on the previous slide. We can see that 1 to 2 cells contain histamine but are not labeled with the Hdc Gal4 RA-RB-RD gene fusion, indicated by the arrowheads. And 3 cells appear to be labeled with the Hdc Gal4 RC gene, indicated by the asterisks, and contain histamine. It appears that Hdc Gal4 RC labels a subset of the cells that express GFP and contain histamine, which does not seem to be the case in this top image, which further indicates a difference between our two Hdc Gal4 hybrid genes. Through our research, we have figured out that there are differences in the expression pattern between the short and the long form of Hdc mRNA in larvae and adult flies. More work does need to be done to examine differences in the adult CNS. The point of identifying Gal4 is to use Gal4 as an activator for other genes that we can express in these cells in the CNS. In our research, we work to identify which Hdc Gal4 flies will be useful for studying the physiology of histaminergic cells and differences in the adult CNS. This research helps identify tools that we can then use for other experiments where we can investigate the function of histamine in central brain cells. To explore the function of these central brain cells, we can use optogenetic tools available in Drosophila. Thank you for your time and attention. If you have questions or comments, feel free to use this QR code.